All right, Mr. Martin's class, buckle up. We're diving deep into cell and nuclear division. You might think it's pretty straightforward, but trust me, it gets wild, especially when you start thinking about life, how it continues, how it changes, all starting at the cellular level.
Absolutely. So in this deep dive, we're going to try to really explore the core processes,
Yeah.
mitosis and meiosis, like what has to happen before these divisions can even occur, the key events in each, and how they are essential to everything, from growth to repair, even to the process of sexual reproduction in eukaryotes.
So like pretty much everything.
Pretty much.
And we've pulled together a bunch of different resources to guide us today. We've got a super detailed guide all about cell and nuclear division. And to back that up, some notes that explain these complex processes. And I know sometimes they can be a little confusing. Well, we're going to try and distill these concepts and ideas into a way that is clear, engaging, and hopefully accessible, especially for those of you who are learning in English as an additional language. So first things first, why is cell division so important?
Well, the cell theory, which is like one of the big foundational ideas in biology, tells us that all living organisms, from the simplest little bacteria to us all, come from pre-existing cells through cell division.
It's like life from
And
life.
from life.
I like it. Okay, so every new cell comes from an older cell that has divided. It's like a parent cell, or what some call a mother cell, that undergoes a process to produce two new daughter cells.
Yeah.
It's like the original cell is making a copy of itself, or splitting, I guess.
Right. And for multicellular organisms like us, these new cells are how we grow, how we develop from a single fertilized egg into, well, us. And it's not just about getting bigger, it's also vital for repairing tissues. You get a cut, your skin heals, the cells in your body are constantly dying and being replaced.
And all of that is cell division.
And all of that is cell division. And of course, for reproduction, especially on a cellular level, we need these divisions to create new organisms.
Right, so we know a cell needs to divide. Now what?
After the nucleus divides, you have to split the rest of the cell, right? And that is called cytokinesis.
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 Got it.
It's the division of the cytoplasm, so all that material outside of the nucleus into those two new daughter cells. Now, here's the cool part. This process isn't exactly the same in all cells. Animal cells and plant cells, for instance, do it a bit differently.
Oh, really? Tell me more about that.
Well, in animal cells, there's this thing called a contractile ring. It's made of actin and myosin proteins, which, if that sounds familiar, it's because those are the same proteins that help our muscles contract.
Oh, interesting.
Isn't it? So this ring forms right under the cell membrane around the middle of the cell, and then it starts to tighten. Imagine a drawstring bag cinching closed.
Oh, I see.
That's basically what this ring does. It pinches the membrane inward, creating what we call a cleavage furrow, and it keeps going deeper and deeper until, poof, two separate daughter cells.
It's like squeezing a ball of clay in the middle until it splits.
That's a great way to picture it. Now, plant cells, they have cell walls, right? So they can't just pinch off like that. Instead, they build a new wall right down the middle, from the inside out.
So how does that work?
They use these things called microtubules. They're part of the cell's internal support structure. They guide these little sacs called vesicles from the Golgi apparatus, kind of like a packaging and shipping center for the cell. They guide them right to the middle of the cell.
Like delivery trucks bringing in materials.
Exactly. And these vesicles fuse together, first making a new cell membrane and then building out this structure called a cell plate. And the cell plate keeps growing as more and more vesicles join in until, boom, it reaches the original cell walls on either side, and voila, we've got two new cells.
So animals pinch, plants build a wall. I like it.
Oh.
Now, what about this equal versus unequal cytokinesis I read about?
Ah, good question. Usually cytokinesis is roughly equal. This makes sure that each daughter cell gets about the same amount of cytoplasm and all the organelles they need.
Okay, that makes sense.
But there's a really important detail here. Each daughter cell has to receive at least one mitochondrion. And for plant cells, they need at least one chloroplast too.
At least one. Why is that so important?
These organelles, they have their own DNA separate from the DNA in the nucleus. And they can only arise from pre-existing mitochondria and chloroplasts. They can't just be built from scratch. So even if the cytoplasm doesn't split perfectly evenly, each daughter cell needs to get at least one of these to survive and function. They need the machinery to make more.
It's like the starter kit.
Exactly. Now, unequal cytokinesis happens in specific situations where the two daughter cells actually have different jobs or fates. One example is oogenesis in animals, the formation of egg cells.
You okay?
When the precursor cell goes through meiosis, which we'll get to later, the cytoplasm divides very unevenly. One daughter cell, the one that'll become the egg, gets most of the cytoplasm and all those nutrients. The other cells, called polar bodies, are much smaller and they eventually break down.
So it's about making sure that egg is well fed.
Precisely. And another example is budding in yeast, a type of asexual reproduction. A small bud grows out of the mother cell, the nucleus divides, one nucleus goes into the bud, and eventually it pinches off to become its own little cell. It's smaller than the mother cell, of course, but it's a whole new individual.
So sometimes it's about making a smaller copy.
Exactly.
Fascinating. So we've got the cytoplasm dividing, but before any of that can happen, we've got to deal with the nucleus, right?
Absolutely. Nuclear division has to happen before cytokinesis. If a cell splits without the nucleus dividing, you'd end up with cells without a nucleus, nucleate cells.
And those aren't going to work very well, are they?
Nope, not for long. Red blood cells are a good example. They lose their nucleus as they mature, and because of that, they only live for a few months. They can't make new proteins, so they eventually need to be replaced. For most cells, having a nucleus with a full set of DNA instructions is essential for survival and function.
So nuclear division comes first. Make sure each daughter cell gets a copy of the DNA blueprint. Then the rest of the cell can divide. And we've got two main types of nuclear division in eukaryotes, right? Mitosis and meiosis.
That's right. Mitosis usually results in 2 genetically identical nuclei, and meiosis results in 4 genetically unique nuclei with half the number of chromosomes. But before either of these can start, something else has to happen first. DNA replication.
Ah, copying the DNA before dividing. Makes sense.
Precisely. DNA replication has to happen during the inner phase of the cell cycle, which is the phase before mitosis or meiosis. So we start with each chromosome being one DNA molecule, and after replication, each chromosome has 2 identical copies called sister chromatids.
So basically 2 identical recipe books joined together.
I like that analogy. And they're held together at a central region called the centromere, and by these protein complexes called cohesion proteins, which act like glue along their length until it's time to separate. And it's not just the DNA that gets duplicated. The histone proteins, which the DNA wraps around to stay organized, also get copied.
So we're doubling everything, the information, the packaging, everything.
Exactly. Now, mitosis and meiosis, even though they have very different outcomes, they also share a couple of common features, like chromosome condensation and movement.
Gotcha. So tell me about that.
So in both cases, the cell has to manage and separate these really long DNA molecules. And to do that effectively, they have to be condensed. It's like taking this long, loose thread and winding it up into a neat little spool. We're going from loosely organized chromatin to these compact, visible chromosomes. And that's where histone proteins come in. They help the DNA to supercoil, becoming much shorter and thicker.
So prepping that cargo for transport.
Precisely. And once condensed, these chromosomes need to be moved to opposite ends, or poles, of the cell. We want to make sure each new nucleus gets a full set of genetic information, right? And that is where the mitotic spindle comes in. It's made of microtubules, which are like the cell's internal scaffolding, and these awesome motor proteins. These motor proteins attach to the chromosomes at a specific spot on the centromere called the kinetochore. They literally walk along the microtubules, pulling the chromosomes where they need to go.
So kinetochore is the handle, microtubules and motor proteins are the movers.
Yeah.
Amazing.
Exactly.
Okay, now let's dig into mitosis specifically, the one that's all about keeping things genetically the same.
So mitosis is, making copies, identical copies. It's the division that gives us 2 genetically identical nuclei. It's what drives growth, development, and repair in us multicellular organisms.
Okay, and mitosis has those 4 main stages: prophase, metaphase, anaphase, and telophase, right?
Yes.
Let's break it down.
All right, let's start with prophase. That's where the cell is basically getting ready to divide. So that chromatin, all loose and spread out in the nucleus, starts to coil up tightly and condense into visible chromosomes. Each chromosome at this point is made up of those 2 sister chromatids joined at the centromere.
Okay.
And at the same time, the nuclear envelope, which is that membrane surrounding the nucleus, breaks down. And the centrosomes, those are the microtubule organizing centers, they duplicate and start moving towards opposite poles of the cell. And as they move, they start building the mitotic spindle, that network of microtubules that's going to pull those chromosomes apart.
It's all about setup.
Exactly. We need the DNA packed up, the nucleus out of the way, and the transportation system in place.
Got it. What about metaphase?
Metaphase is all about organization, alignment. The spindle microtubules extend from those centrosomes and attach to the kinetochores on each chromosome. And through this balance of pushing and pulling forces, the chromosomes get lined up right in the middle of the cell. This is the metaphase plate, and it literally looks like a perfect line of chromosomes in the middle of the cell.
It's beautiful.
It is.
Okay, so they are lined up, then what?
Anaphase. Think apart.
Makes sense.
Anaphase is when those sister chromatids finally separate. The cohesin proteins holding them together get broken down, and now they can be pulled apart. The microtubules shorten, drawing the chromatids, which we now call daughter chromosomes, towards those opposite poles. And the cell itself elongates as the spindle poles move further apart.
So the identical copies are separated and heading to opposite sides of the cell.
Exactly. Then we move into telophase. Think the end. This is where we reverse some of what happened in prophase. So those daughter chromosomes arrive at their poles, and a new nuclear envelope forms around each set. The chromosomes relax and uncoil back into that loosely packed chromatin, and the mitotic spindle, it disassembles. It's done its job.
Wow, that's quite a process.
It is.
So now we have two nuclei each with a complete set of chromosomes, just like the original.
Exactly. And then we move into cytokinesis, and we get two separate cells, each genetically identical to the parent cell. That's mitosis.
And mitosis isn't just about making more cells for growth, right?
No, it's much more than that. It's how many eukaryotes reproduce asexually, making genetically identical offspring.
Wow.
It's how somatic cells, the cells of our body, grow. And as we talked about earlier, it's crucial for tissue repair and regeneration. So your bones grow, plants grow taller and longer, all thanks to mitosis. Skin renewing itself cuts healing, all mitosis.
Incredible. And the mitotic index, can you explain that?
Yeah, it's a way to measure how actively cells are dividing in a sample. We look at the number of cells that are in any stage of mitosis and divide that by the total number of cells. Higher mitotic index means more cells are dividing. And this can actually be a clue for things like cancer.
Oh, really?
Cancer cells divide a lot faster than normal cells, so a high mitotic index in a tissue sample can be a warning sign.
So understanding cell division can help us diagnose and understand diseases. That's powerful.
It is. It's not just theory. It has real-world applications.
Okay, on to meiosis, the one that's all about generating genetic diversity.
Okay, so this one's a bit different. It results in 4 haploid nuclei that are all genetically different from each other and from the original cell.
Haploid meaning
Half the number of chromosomes.
half the number of chromosomes. Okay.
Meiosis is essential for sexual reproduction in eukaryotes. It's how we make gametes, sperm and egg cells. By reducing the chromosome number by half, we make sure that when fertilization happens, the offspring end up with the right number of chromosomes.
So you don't keep doubling the number every generation.
Exactly. Meiosis is 2 divisions, meiosis the second and meiosis the second, and each one has those 4 phases: prophase, metaphase, anaphase, and telophase, followed by cytokinesis.
All right, let's go through them. Starting with meiosis, the one that separates homologous chromosomes.
Okay, so in prophase I, the nuclear envelope breaks down, the spindle forms, the chromatin condenses into chromosomes that part similar to mitosis. But something really important happens here that's different. The homologous chromosomes, so the pairs, one from your mom and one from your dad, they come together and pair up. This is called synapsis. They form these little bundles called bivalents.
So it's mom's copy and dad's copy, paired up.
Yep. And while they're paired up, they do this incredible thing called crossing over. Crossing over. The non-sister chromatids, so one from mom's chromosome and one from dad's, they swap segments of their DNA at random points called chiasmata.
So they're like swapping recipes.
Yes. They're trading alleles, different versions of genes. And this means those sister chromatins are no longer identical, and it introduces a lot of genetic variation in the resulting gametes.
It's a mix and match.
It is. And it's super important for evolution.
Yeah.
It gives us all this new material for natural selection to work with.
Okay, so after this recipe swap, what happens in metaphase I?
Now, those bivalents, the pairs, line up along the middle of the cell. But unlike mitosis, where single chromosomes line up, here it's the pairs that line up, and each chromosome in the pair is attached to microtubules from only one pole of the cell. And importantly, the way they line up is totally random.
So it's random which side mom's chromosome goes to.
Totally random. And it's independent of how the other pairs line up. It's called independent assortment. It's like having several pairs of shoes, and you can arrange them in all sorts of different combinations. Same with the chromosomes. It all leads to lots of different combinations of mom's and dad's chromosomes in those daughter cells.
So much variation.
So much variation.
Okay, so then anaphase tally happens.
Yeah. Yes. Now the homologous chromosomes get pulled apart. So mom's goes one way, dad's goes the other way, but the sister chromatids, those stay together for now.
So the pair is split, but each chromosome is still double.
Exactly. And then in telophase 1, those chromosomes arrive at the poles, the nuclear envelope might reform, and cytokinesis usually happens. We end up with two haploid cells. They're haploid because they only have one chromosome from each pair. But each chromosome is still doubled. It still has its sister chromatid attached. And importantly, there is no DNA replication before meiosis II starts.
Okay, we have the number of chromosomes, but they are still doubled up. Now on to meiosis II, which separates those sister chromatids.
Exactly. And meiosis II actually looks a lot like mitosis. So in prophase 2, the nuclear envelope breaks down again, the spindle forms. In metaphase 6, the chromosomes, still with their sister chromatids, line up in the middle. But this time, each sister chromatid is attached to microtubules from opposite poles.
So now it's back to single file.
Yep. And in anaphase 2, those centromeres finally separate, and the sister chromatids, now we call them chromosomes, head towards opposite poles.
And finally, they are separated.
Finally separated. And in telophase 2, they arrive at the poles, new nuclear envelopes form, the chromosomes uncoil, and cytokinesis happens.
Okay, so what's the final product of all of this?
We end up with 4 genetically unique haploid daughter cells. In animals, these will become gametes, sperm, or egg cells. And the uniqueness comes from that crossing over and an independent assortment that happened back in meiosis I. Think about it. If we just look at independent assortment, if an organism has N pairs of chromosomes, there are 200 possible combinations in the gametes.
That's a lot of combinations.
It is. In humans, with 23 pairs of chromosomes, that's over 8 million possible combinations in each sperm or egg, and that's before we even factor in the diversity from crossing over. That's the power of sexual reproduction.
Mind-blowing. So all this is super precise. What happens if something goes wrong? I read about non-disjunction.
Ah, yes, errors can happen. Non-disjunction is when chromosomes fail to separate properly. It can happen in anaphase I or anaphase II, and it leads to some gametes having an extra chromosome and some missing one.
And this can have big impacts on the offspring.
Definitely. A common example is Down syndrome, also called trisomy 21. It's caused by an extra copy of chromosome 21. So the individual ends up with 47 chromosomes instead of the usual 46.
So one tiny mistake can have a big impact.
It can.
Okay, so we know how cells divide, how DNA is managed during all of this, but what's controlling the whole cycle? It's not just happening randomly, right?
No, it's very tightly regulated. The cell cycle, as we call it, is this ordered sequence of events a cell goes through as it grows and divides. And this is essential for normal cell growth.
And the cell cycle has its own phases, right?
It does. We've got interphase, which is all about growth and preparation, and then the mitotic phase, which is the actual division, mitosis, and cytokinesis. Interphase is actually the longest part. It's divided into G1S and G2. During G1, the cell grows and does its normal thing. In the S phase, DNA replication happens. And then in G2, it keeps growing and preps for division.
So it's living its life, then prepping to divide, then actually dividing.
Exactly. And it's all very carefully controlled by these proteins called cyclin-dependent kinases, or CDKs, and cyclins.
And what do they do?
Well, CDKs are enzymes that can activate or inactivate other proteins by adding these phosphate groups to them. We call it phosphorylation. But CDKs can only work when they are bound to a cyclin.
So cyclins turn the CDKs on.
Exactly. They are like the key that starts the engine. And different cyclins are made and degraded at specific times in the cell cycle, so their concentrations go up and down, controlling when specific CDKs are active. It makes sure everything happens in order.
So it's a timing mechanism.
It is. And there are also these checkpoints along the way at G1, S, and G2 where the cell basically checks to make sure everything is good to go before moving on.
So it's double checking its work.
Exactly. Making sure the DNA is okay, that there are enough resources, and certain cyclin CDK complexes need to reach a certain level to pass these checkpoints. Like, okay, we're good, move to the next stage.
So what happens if this control system breaks down?
That's when you get things like cancer. Mutations in genes that control the cell cycle can be really bad news. Proto-oncogenes, these are normal genes that promote cell division. Think of them like the gas pedal. But if one mutates, it can become an oncogene.
An oncogene.
And this oncogene can be stuck on, leading to uncontrolled cell division.
Oh no, a stuck gas pedal.
Exactly. And then you have tumor suppressor genes, which are like the brakes. They normally slow down or stop the cell cycle, maybe to repair some DNA damage or to trigger cell death if the damage is too bad. But if a tumor suppressor gene gets mutated, it's like losing the brakes. Cells with damaged DNA can keep dividing.
So whether it's the gas or the brakes that break, you can end up with uncontrolled growth.
Exactly. That's how you get tumors. They can be benign, which means they grow slowly and stay put. Or malignant, which means they grow fast and can spread. That's what we call cancer.
And spreading is what makes it really dangerous.
Yeah, that's metastasis.
Yeah.
The cancer cells break off and travel to other parts of the body.
Scary stuff.
It is. So before we finish, there are a couple other ways cells can divide, right? Besides mitosis and meiosis.
Oh, right.
So prokaryotes, like bacteria, keep it simple, they use binary fission. They don't have a nucleus. Their DNA is just this one circular chromosome. So they replicate their DNA, and then the cell splits in two. No spindle, no complex chromosome movement.
Super efficient.
Very efficient.
What about budding? We talked about that earlier.
Yeah, that's how some fungi, like yeast, reproduce asexually. But some simple animals do it too. They grow a little bud, the nucleus divides, one goes into the bud, and then the bud pinches off. The cytoplasm usually divides unevenly, so the bud is smaller than the parent cell, but it's a new individual.
It's fascinating how many different ways there are to divide a cell.
It is. So to sum it all up, mitosis is for growth and repair, keeping things genetically the same. Meiosis is for creating genetic diversity for sexual reproduction. Both rely on DNA replication and those carefully choreographed chromosome movements. And the whole thing is controlled by this intricate system of checks and balances. And when that system breaks down, you can end up with uncontrolled cell division and things like cancer.
Life is complicated.
It is, but amazing too.
Okay, Mr. Martin's class, I really hope this deep dive has helped you understand cell and nuclear division a bit better. It's definitely more than just memorizing the steps, right? It's about understanding how life continues, how it changes, how it creates new possibilities. If you found this helpful, please like and subscribe to our channel. We've got more deep dives coming your way. And if you have questions, leave them in the comments below. We'd love to chat.